Assalamualaikum
Assalamualaikum
warahmatullahi
warahmatullahi
wabarakatuh.
wabarakatuh. My name is Aisyah Fadhilah and this is my partner Muhamad Nabil Alhanif. We are from SMA Kharisma Bangsa. Today we're going to present our project called C-ATP, Computational Analysis of Aloe vera for Transgenic Insulin Production.
So in the top left corner, there is a bar graph that shows the number of diabetics in the top 10 country with the most number of diabetics in the world in 2010 and 2030. And we could see that by 2030 there will be a significant increase in the amount of diabetics in the world. Therefore, it's predicted that the amount of insulin needed will rise by more than 20%, and half of those in need will lack access. Uh, this insulin shortage will definitely raise the price of insulin, which will make people in poor country harder to get insulin. So we need another alternative to cope with this high insulin demand in the future with a more effective approach.
The objective and goals is that generating computational data and virtual experiment as a base for further in vivo and in vitro research in creating transgenic aloe vera plant properties for insulin and also as comparison for real-life data. The limitation is that the experiment is an in silico design and have been tested for in vitro experiment yet. The novelty is that the pro-insulin production in transgenic plants have been researched before, such as transgenic tobacco mediated by agrobacterium tumefaciens. While in this research, we offer an in silico approach to producing pro-insulin within the chloroplast. These are our literature review. In this experiment, we use the in silico because it's need a shorter period of time and also the cost relatively less than in vivo and in vitro. These are the picture of insulin, and you can see the conventional method for transgenic insulin using E. coli. These are the advantages and disadvantages of it. We can see the current meta problem from this table using the E. coli. Therefore, we offer another alternative based on biofarming and also using the transgenic plants because there are so many advantages of it. One of them is there are post-translational modification.
And we choose aloe vera because of some consideration. The first one is the ease of extraction, as the insulin will be localized in a specific area, and aloe vera also has high production rate and can be cultivated in many places, and aloe vera also has post-translational modification. And here's the map of aloe vera distribution, and here's the data of aloe vera cultivation and production from Pontianak.
These are the expression system. We use stable expression using the particle bombardment chloroplast as a target. This is our methodology.
So we use some software and data resources as shown in the current slide. And at the top image there is the image of the human proinsulin from GenBank, and the bottom one is optimized human proinsulin for aloe vera. And for the codon optimization itself, we use the codon usage table of aloe vera chloroplast from Kazusa database species 34199. And after that we do pairwise alignment to compare the sequence before and after the codon optimization. Then we start assembling the vector. The first step in assembling the vector is designing the primer and doing PCR. Our primer is designed automatically by Snapkin for PCR, while the restriction site are added manually with data from the restriction mapper as consideration. Next step is to do viral digest and electrophoresis, as shown in the current slide. And we do viral digest and electrophoresis to predict the length of our gene of interest. And the last step is to do cloning. So we use Snapkin to simulate insertion cloning with the help of T4 ligation enzyme. The vector we use for cloning is Picambia 1300 cloning vector.
We use the particle bombardment method due to the double membrane of the chloroplast that would be difficult to conduct with any other method. These are the pros and cons using the particle bombardment, and you can use the in vitro experiment using this procedure. And these are our data visualization and analysis.
So the next step is to generate a PDB file, which is basically a protein file for the visualization of the protein. We use Tropeda Baker Lab to visualize and generate the PDB file, and we use PyMol to analyze results. In total, there is 4 different protein models generated by Robeta, which we'll show in later slides. And please note that Rhombithiolite is evaluated continuously by Chemio, and protein structure prediction is based on other similar protein. And currently, there is no perfect method to predict protein output and structure, which may explain why real-life experiment results may differ from in silico results.
And these are the first model and explanation of it. And this is the second model. And the next one is the third model. And this is the fourth model. Conclusion. Aloe vera is suitable as transgenic plant to produce insulin because it has been researched by many scientists, especially for the transgenic protein itself. And the aloe vera offers various advantages, including ease of multiplication, extraction, making it more economical, and lowering the risk of contamination by toxic substances, and requiring relatively short time with high production yields, thus increasing productivity, especially in tropical regions such as Indonesia.
And through the data we gathered from Penang Agricultural Department, aloe vera also have a high rate of production, thus making it a promising natural and eco-friendly insulin production factory. Our in silico experiment had also shown that aloe vera has a capability to produce insulin, although more tests is needed to measure the effectiveness and efficiency of transgenic aloe vera insulin.
These are the references that we used. And thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.